lesson 17 is just this titled Unlocking the Code. And it's specifically because, as I keep saying to you guys over and over again, DNA is just a big, long, DNA is just a big, long code. It's just got a lot of, C, A, G, T, whatever. It's just a big, long code. But how can we take a big, long code and get to something like a bird or, a mouse or whatever? How can we turn, that's a mouse. How can we go from this code into this? And that's the really big part. And that's the part that a lot of people don't know. People will say to you, oh, DNA is, everyone's, is the blueprint. It's like, yeah, but how do you go from blueprint to like an actual, building? That's the real question we have here today. Jumping back for a second, I'm going to go backwards. I want to look at what we did last time. Last time we looked at DNA, we actually looked at its structure. We said that DNA is made up of two bridges, or two rungs. And in those two rungs, we have we have some rails, and on those rails we have we call those rails a backbone, I suppose. And on that backbone we have two parts, a phosphate and a sugar. And these phosphates and these sugars repeat each other over and over and over again. And then attached to the sugar is a base. And we refer to this whole thing as a nucleotide. So we've got heaps of nucleotides that go up and down the DNA string. And on one side, you've got all these phosphate sugars. On the other side, you've got all these phosphate sugars. In the middle, you've got these bases. And the bases form, the bases form four different bases: adenine, thymine, cytosine, and guanine. Now, what's special about these bases is that they fit together. A fits with T and C fits with G. And because we know that these bases fit together, we know we can then match different things. We know that a T is only going to bond to an A. We know that a C is only going to bond with a G. We know that a T is only going to bond with an A. We know an A is only going to bond with a T. We know a G is only going to bond with a C. So we can actually figure out what's on the opposite side of a DNA string without even looking at it, because we know that they have to fit. We can also do some maths, and I talked about that just because we said that if we know that there are 10, let's just say we know there are 10 G bases, then there must be 10 C bases. That's just how it works. If we know that there are 5 A bases, then there must be 5 T bases, so on and so forth. So let's talk about today's lesson. And I wanted to start with this big question, okay? On the left at the top, I've got a picture of a blueprint for a building. And on the right, I've got a picture of that building. Now, we're happy saying that DNA is linked to the blueprint. DNA is like the instructions that you need in order to make a building. An organism, like a bird, is like that building. But the real question here today is, how do I get from the left to the right? How do I take the list of instructions and how do I make that into a building? I mean, how can you read this complex code? How can you take this complex code and turn it into, turn it into something that will actually exist? And that's what we're going to be looking at today. Now, our very first step in understanding how we go about this is talking about one of the molecules that makes this connection possible. And that molecule that makes this connection possible is RNA. Now, RNA, and this is not a typo, is called, or ribonucleic acid is another type of nucleic acid. So there are actually different types of nucleic acid. DNA is deoxyribonucleic acid, whereas RNA is just ribonucleic acid. Let's talk about some of the differences between RNA and DNA. First one, it only has one backbone. If we look here, it's only got one side. It's like DNA, but it's a one-sided DNA. So it's a bit different in that way because these bases, instead of attaching to anything, kind of float around in whatever. It's the backbone though is still it's still phosphate sugar all the way down the backbone. So don't stress that way. It's still pretty similar, and the bases are pretty much the same. It uses all the same bases, except there's one other exception. Instead of thymine, we use uracil. Now you might say, well, what the heck is uracil? So if you look at this, we've got cytosine, cytosine, adenine, adenine, guanine, guanine, thymine, uracil. So you might be like, well, okay, what does that do? If you remember that AT means, you know, apple tree, and GC, or CG means car, garage, you might just want to change one of them to be, you know, under the tree. That's why we call it UT. Or you might have a different way that you remember it. It's just a new base. Now, the code in RNA is broken up, is read by looking in 3 bases at a time. So, and a 3-base section is called a codon. I'm going to talk a little bit about that now. Let's just draw a random piece of RNA down the bottom here. So, and these things here, we're going to call these the bases. So 1, 2. I'll stop about here. So how a codon works is it looks at 3 bases at a time. So 1, 2, 3, that's a codon. 1, 2, 3, that's another codon. 1, 2, 3, that's a third codon, and so on and so forth. So codons are groups of 3 bases, and there's only a certain amount of combinations of 3 bases that you can have. This picture at the top is sort of trying to link together the idea. And this is a good place to sort of, you know, link everything together. In this picture, I have a man and a woman that are both looking at a blueprint. One of them looks like a contractor and one of them might be an architect. But the idea is these guys are looking at the blueprint to build the building, okay? These guys are decoding what's on this blueprint so they know what they need to build. So that way, in that way, these guys are taking this blueprint and they're moving to building the building. So the question then becomes, yeah, so that's what RNA is for. RNA is really important in this link. I haven't said why it's important yet, but I've said it is important. And we're going to start now to talk about, well, how does RNA, what does it do for the DNA? What it does for the DNA is that there is a section here, there are 2 new words we need to learn. They are called transcription and translation. And that's what we have to really learn today. They're a bit of complex topics, but I'm going to talk about what they mean. Transcription is all about moving from DNA to RNA. It's about taking the blueprint that we've got in our DNA molecule and turning that into RNA. It's about de-transcribing the information, so copying the information. Whereas translation is all about taking the RNA and turning that into proteins. I don't know how to draw a protein. We're going to draw a big, long, flat thing here, and I'll talk briefly about that. So we're going to talk about each of these two topics individually. Transcription first and translation second. Okay, I haven't put a pencil on this slide. I suppose you don't need to copy this down. This is just something to get you guys familiar with what we're going to talk about. We're going to start with transcription first, and then we're going to do translation second. Transcription, what does it mean? Okay, this next slide's got a fair bit of text on it. Okay, so transcription. I'm going to take some time here. If I start speaking too quickly, let me know. Transcription. What does transcription mean? To transcribe means to copy something down. So if I said to you, I want you to transcribe this this PowerPoint page, it means I want you to write this PowerPoint page down. And you might even see the word scribe in this word. You guys may have seen that word scribe before, because usually when we get people to write down for a group project, we say, hey, can you be the scribe? Scribe means to write down. Transcribe means to go from one place to another. So what does transcription, transcribing, what does that mean in this context? Let's take a strand of DNA. Here is a strand of DNA. Okay, all the T's match with all the A's, all the C's match with all the G's. They're all connected. Everything's happy. Yeah? So what's the first thing that happens is the DNA unzips down the middle. Now, when we say the DNA unzips down the middle, what we actually mean is pretty much all of these bases separate from each other. Okay? The T's separate from the A's and the G's separate from the C's. Now we can easily zip them back up together. Okay? We could just push them back together quite easily. All I'm doing is just pulling the two sides apart. The reason we call this unzipping is because it's a little bit like a zipper on like some pants. When you unzip something, they spread apart, and then you can zip them back up, and you can zip them back up together. So the first thing that happens is you unzip down the middle of the DNA. Okay? Okay, the second thing that happens, number 2, is then what happens is pieces of RNA. Now we said that RNA is a small string, as a string of bases. RNA move into the selected parts of the DNA to make messenger RNA or mRNA. Let's see how that works. Let's see this actually happening. So inside the nucleus, this is all happening inside the nucleus of the cell. We've got these random parts of RNA zooming in. This here is mRNA. M standing for messenger. So messenger RNA. And that's because this messenger RNA is bonding. Now let's grab a look at this as messenger RNA. Notice how that there's an A bonding with a T, because that's what we know happens. We've got a C bonding with a G, because we know that happens. And we've got a U bonding with an A, because remember, we don't have T anymore. We have U now. This messenger RNA is very important. This messenger RNA has essentially made a copy of this side of, if you look, this messenger RNA has made a copy of this side of the DNA. TCG, TCA, UCA. It's pretty much a copy. So it's transcribing, it's copying down part of the DNA sequence. Am I going too fast? Am I going okay? Have I lost anyone yet?
Oh, good.
No.
We're good?
Yep.
Okay.
Yeah.
Cool. I'll keep going then. Let me know if you're like, whoa, 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 what's going on? Okay, so we've copied down half of the DNA. When I copy down half of the DNA, this part of the DNA, I'm gonna now move that DNA, I'm gonna move that RNA out of the DNA. I'm gonna zip the other side back up. Okay, so now I have a copy of half of the strand of the DNA. Now, of course, obviously in real life, well, maybe not obviously, in real life, this RNA string would go for a long time. It would just go for 3 things. It'd probably go for like, it could go for as many as, 100, 200 bases or something. It'd be a lot longer. But I'm just sort of showing you here, this is just a small sample. So what I'm going to do now, hold on a second, my dog's butt. Juno, shush. Sorry, guys. So now the mRNA has a special property. This is the mRNA here. Okay. I do want to say, by the way, you guys may never have seen RNA before, but RNA is actually older than DNA. People actually think that organisms had RNA way before they had DNA. So I'm going to remodel these. Instead of drawing a big long DNA string like this, an RNA string like that, I'm going to draw it like this. Think about it. I've got one string with a couple of bases, a string with a couple of bases. And now I'm going to model, instead of this big long DNA, I'm going to use this as a model. Are we comfortable with that? Me going from those pictures to these pictures? Does that make sense?
No.
Yeah.
I get it.
Kind of.
Okay, let's have a look. Let's have have fun. So we've got, all I'm doing is instead of drawing the, this big long string here with these bases, all I'm doing is I'm drawing this string with some bases. These here at the end, those are the bases. And this here, this is the backbone. Okay? Instead of this big long ladder here, all I'm doing is I'm going to replace it with this picture here. But what I might do as well is I might label that. That's the DNA. This is the mRNA, the copied down RNA. Now, the third part of this is, this part here. So this is all happening inside the nucleus. Now, what makes, and I'll re-put the labels here. Here's DNA, here's the mRNA. Now, what makes the mRNA?
Do we copy the diagram or no?
What I would like you to do, is I would like
Just on Google.
It's gonna be, you guys are gonna be able to access this afterwards. I think what I would like you to do is maybe not copy down the whole diagram, but just sort of like some pictures. So maybe you could say, one, it doesn't have to be exact. So for one, you would say, I would unzip. So see how I've just done the same picture as I did at the start, but I've simplified the picture. And then 2, I would then draw this, and then I would draw a new bit of DNA in the middle, a new bit of RNA, sorry, in the middle. And then for 3, I would draw this slide here. So I'm just going to draw a bubble kind of like that and like that. And that's how I'm just trying to draw simple pictures, not obviously as complicated as what I'm doing, but I think you should have. And if you go 1 goes to 2, then 2 goes to 3, whatever. But I'll quickly finish explaining it first. The next part is this. The advantage of the mRNA, the messenger RNA, is it's very small. And because if you compare it to the big, long DNA, it's just a tiny, tiny little bit of information that's being copied. So because it's a lot smaller, it can actually escape the nucleus. It can actually fit through holes in the nucleus. So this is like a hole or a pore in the nucleus. The DNA is too fat to fit through, but the RNA is just small enough that it can fit through. And when that RNA comes out of the nucleus, it goes into something. The mRNA enters another part of the cell called the ribosome. So the ribosome, which I've got over here, is just another part of the cell. You don't need to know exactly what that is right now, but you do need to, know what it's called. So yeah, I guess these are the 3 steps. Firstly, you unzip it, then you copy it, and then you move it out of the nucleus. All right, so are there any questions about that?
What do you unzip?
Okay, the DNA gets unzipped. So watch. So
like that.
As I unzip it, so I'm going to move each of these each of these bases apart. This is how it unzips. Yep. Then after it unzips, I copy 1/2. Then I re-zip it back up and I move my copy out of the way. Then, because that copy is small enough, I can move that copy out of the nucleus.
So the whole point of transcribing is to make something small and just move it.
Yeah, the whole point of transcribing is to make a copy of it and get it out of the nucleus. So we're just making a copy of it. Imagine that, imagine, this is going to be a really bad analogy and I apologize, but imagine that I send you to the library and I'm like, okay, guys, I want you to find out what's on page 45 of the encyclopedia. And the encyclopedia is like this massive book. It's just huge. And you're like, whoa, I'm so small, I can't possibly pick up this whole big book. What you might do instead is I'm just going to look at page 45 and I'm going to write down what's on page 45 and then leave the book there. And then I can go, I can go home. That's exactly what's happening here. Okay, maybe not exactly, but that's sort of what's happening here. The messenger RNA is copying not the whole DNA strand, just a little bit of the DNA strand. And then it's leaving the nucleus, it's leaving the library with that information. How about that? What I'm going to get you to do for me is this. I'd like you to, in chat or out loud, up to you, I want you to give me a number, 1 to 5, to tell me how much you understand what's happening here. 5 meaning that you get it so well you could teach someone else. 1 meaning, okay, I recognize some of the words. 2 meaning, yeah, I get it, sort of. 3 meaning, I get it, but I might need some help. And 4 meaning,
3.
I actually understand this. I just don't think I could explain it to anyone else.
3.
Thanks, David. Yep, thanks, Alethea. Cool.
He said 7.
Wait, so did you say out of 5?
Yeah, 5 meaning that you get it so well you could explain it to someone else. 4 meaning you get it, but you couldn't explain it to someone, but you get it. And 3 meaning I get it, but I'm going to need some help probably.
Wait, so what happens if you can explain it and with enough time they'll get it, but it's a bit sloppy?
I would say 5 then. If you're confident enough that that you can explain it to someone, if you're confident enough that you could explain it to someone and teach it, then I'd say that's 5. And that's, that's pretty good. Like that means, hey, and even, yeah, sure. Like obviously we're not all going to be perfect here. I am not a genius when it comes to describing this. I didn't get a chance to actually learn about this when I was in high school. So I was really confused. I'm like, I have to teach transcription and translation. Jeez, I never learned this stuff. Okay, so I've got a lot of 3s and 4s and a couple of 2s, which means that I'm going to keep moving on. And I'm going to explain later on why it's okay that you've got a couple of 2s. So this is transcription. This is the first half. The next half is what happens inside of that ribosome. Okay, so let's talk about that. Hold on a sec. Okay. The next part is called translation. Now, what does translation mean? Translation means to convert information from one type to another. So if I'm trying to translate something from English to, I don't know, French, I'm taking it from one language code to a different language code. If I'm trying to translate something from, I don't know, Latin to English, and taking it from one code to another code. So that's what translation means. We now have, we've got this mRNA, right? And this mRNA, which has gone into the ribosome, that mRNA, which is inside the ribosome, it's kind of still DNA. It's still a code, right? It's still, so this is going to be our ribosome here. We need to take that mRNA and turn it into something. So what we're going to do is we're going to take, okay, so here we've got this big, long, red thing here. This is our mRNA, our messenger RNA. And remember that messenger RNA is still a big, long code. It's pretty much exactly the same as the DNA. It's just still a big long code. So we've still got a U, A, G, C, T. No T's. Why am I writing T's for? We got rid of the T's. A, C, G, U, A, G, whatever. T. No, not T. A. That'll do. What we do is inside the ribosome, we have a new type of RNA. Inside the ribosome, the message is translated, like taking that information and changing it, decoding it using transfer DNA or tRNA. What tRNA does, I'll show you what tRNA is going to look like. It looks like this. And okay, at this point in time, my pictures are not great. tRNA collects the specific amino acids which gets built to make a protein. Proteins such as enzymes, and I'll talk about that. So what this tRNA does is you'll see the tRNA has an amino acid attached to the top. And that tRNA can read parts of the messenger RNA. And it reads, remember at the very start, I said it reads 3 bases at a time. And it reads 1, 2, 3. It reads 3 bases at a time. And when it reads those bases, if it sees the 3 bases that it's looking for, let's just say that this tRNA is looking for UCC. When it sees UCC, it will release a particular amino acid. In this case, and that amino acid will just float around. And then what will happen is that the next three, so we might say this three here, that might release a new type of amino acid. And the next three will release a different type of amino acid. And then all those amino acids, right, will join together to make a protein. Okay, so essentially what the ribosome is doing and what is it's changing this code into a protein. And it does that by reading the code using tRNA. Transfer, oh, that's a mistake here. Transfer RNA, not transfer DNA. And it will change it. And every time it reads it, it'll build up a new amino acid. Proteins such as enzyme. Now, what are the proteins? What are proteins? If you didn't know what a protein was, a protein is like, firstly, an amino acid are the building blocks of proteins. And a protein is pretty much one of the most important things our bodies need. So, for example, enzymes like hemoglobin, really great. They move oxygen around the body. Proteins like insulin, we need them. We've got enzymes, digestive enzymes. They're in our body that we really, really need. Hormones like testosterone and like estrogen and progesterone and yeah, these are all, and of course insulin. These are glucagon. These are all really important parts that we need in order to regulate our body. And of course, proteins can just then generally go towards, proteins can also lead to building up different systems, different organs, and then, you know, eventually different cells and so on and so forth. So proteins are really important when we talk about how to build the human body. So they're the building blocks. So we need to make sure that we have this transferred DNA. So transcription is about copying the DNA. Translation is about turning that code into a protein using tRNA. Now, I'm going to come back to this idea in a little bit, but I'll move on for the moment. I wanted to talk about those codons. So I said they come in 3 letters, right? So those 3 letters might be TAT or GAT or ACT. And those mean different things. Some codons code for the start of the code. So for example, you might have 3 letters that say, yep, this means I want you to start reading the code. Sometimes you have 3 bases that say, I want you to stop building the code. But most of the particular codons, most of the particular 3-letter codes actually just make building blocks of proteins. They just, you know, make amino acids. For example, GAT, or in this GAT or GAU, codes for aspartame. Aspartame. That is an amino acid. So if the tRNA sees GAU, they will make aspartame. That's how it works. Let's have a look to see, so how does the DNA know, how does the tRNA know what goes with what? Well, there are actually these, you don't have to learn this, I just wanted to show you. I wanted to show you guys, this is a bit of a table that how you can work out what amino acids go with what. So let's pick one. We've got the example that we used at the start is we used UCC. How do you use this table? You don't need to know this too much. If this part gets confused, you don't stress. Well, the first letter is a U. So that means we go, okay, the first position is a U. The second position is a C. So that means we're going to go UC. So then it's one of these four. And we've got UCC. So here's UCC. That means it's going to make a particular amino acid called serotonin, or I can't remember what SER stands for, but we'll look it up in a second. Here we can see this one, these are the stop commands, and then we've got AUG. I don't know where the start commands are. I thought ACT was a start command, but I guess I'm confused. That's all right, don't worry. So now, almost done. Here is all of the amino acids. So just in case you're like, okay, what are they? So we said here, SER on this picture is here, serine or serin or whatever. So that's the particular amino acid that will get added to our DNA string next. No, you're not supposed to memorize those. God damn, that'd be really hard and not necessary. Or the transcription and translation is really important. Memorizing how to use. I'm going to get you guys to maybe have a try at this, copying stuff down and figuring out what amino acid's going to get made. But I don't need you to know how to do that. It's not essential. Finishing up, this is just going to be a little bit of a different sort of take here, but okay, we now know that every cell has the full DNA structure.
So are you able to go back to the codon slide?
This one here?
Like the one that we have to take notes of?
This one here.
Yeah.
Yeah.
Thank you.
That's all right. I'll leave it up here for a second. It gives me a chance to quickly drink my coffee.
But what is TAC? I don't even understand what TAC is.
Oh,
No.
that's 3 bases. So TAC, thymine, adenine, cytosine.
Oh, is that like a thing that already exists? Is that like a thing that already existed?
Yeah.
Oh.
So they're just 3. So if I was looking at this over here, right, it could be TAC. That's what I'm talking about when I'm talking about TAC.
Oh, so it's basically anything but with those 4 bases.
Yeah. You could have any of those bases, right?
Mm-hmm.
For example, like, I mean, Brendan, give me 3 bases. Give me any 3 bases.
CAG.
CAG?
Mm-hmm.
Okay. So if I jump over here, CAG, the first letter was C. The second letter was A. The last letter was G. That means it's going to make this particular amino acid, GIN.
What's that?
Well, I can then look that up. GIN. Let's see. GIN. It's not glycerin. It is glycine. Where is it?
I'm pretty sure that was an L.
I'm looking for it. Was it an L?
Yeah.
Oh, it's
L.
GLN. GLN, let's see.
It's in the bottom right.
Bottom right? Beautiful. There it is. Glutamate.
Walter Link.
Glutamate.
Wow, rocket science.
So it's like decoding something.
Okay, cool.
So finishing up, I want to quickly talk about on-off switches. So every cell has the full genetic code. However, here's a bit of a weird thing. Your hair cells, so the cells in your hair, in your head that grow your hair. They don't need to make teeth. They don't need the DNA. You know, you have a part of your DNA that says, this is how you make teeth. Your DNA in your hair doesn't need to know how to make teeth. Your cells in your fingernails don't need to know how to make teeth. Your cells in your feet don't need to make teeth. The only cells that need to know how to make teeth are the cells in your gums. So what your body does is it switches off those genes. So ultimately what my cells do is they say, well, I'm not interested in making teeth, so I'm not going to read this part of the DNA. It's not useful to me. And that's why when you look at people, they don't have like fingers growing out of the back of their neck, because the back of their neck, the cells in the back of their neck don't care about growing fingers. They don't have, you know, hair growing on the inside of their stomach, because the inside of their stomach cells don't care about growing hair. When you see genetic mutations, it's usually because, yeah, some cells somewhere in the body have freaked out and started to do the wrong, reading the wrong part of the DNA. But that's basically what an on-off switch and how it sort of works. Yeah, last bit here. I just wanted to quickly go look at this kind of example that is on style. This does a really good job, this video, of explaining how the essentially firstly how we can go from the DNA structure all the way up to how we can then create a protein, which is this picture on the right. I actually kind of like this part, and they do a lot better of a job of sort of their pictures, because obviously mine aren't too great. But if I grab a look at, I like this part here. So we'll just play from here. I'll keep it on mute so you can hear me, so you can watch the RNA. The RNA is coming out of the nucleus of the cell and goes into the ribosome. And each time a part of that ribosome gets read, it actually creates part of the DNA, the amino acid, and each part of the amino acid then joins to make up a new part of the protein, and that protein starts to coil up and get complex and spin around, and that's essentially because it's, you know, a big long chain. So each of those pieces of RNA are getting read and then making unique, you know, different types of they're making unique parts of the protein structure, and then eventually you end up with this big protein at the end, which goes off to do some kind of operation, like even make a new cell. This is another example here of just this very this is part of the YouTube video that I've asked you to look at. You can see as the tRNA comes in, it clicks into specific parts of the RNA, and it'll click in, so this part will stick onto this part here, and then the next, they are in triplets, there are 3, and that's called a codon. But like, I like this picture here, so they sort of fly in and they'll switch on, and only one will fit. So you can see that only the UAC will fit onto that AUG, and then that will release a specific kind of amino acid. And I think if I look at this part, then the next one will join to that original amino acid, and it will just build up the structure like I showed you in that last video. Anyway, hope that helps. Let me know if you've got any issues, send me an email. Otherwise, good luck. You've got your homework that I want you guys to do, and you should be okay. So this is Mr. Greer signing out.